Good afternoon, everyone. Thanks for joining us today for Infectious Diseases Grand Rounds. I'm going to go ahead and introduce microbiologist extraordinaire, Dr. Alan Junkins. Dr. Junkins, take it away.
Oh, great. Thank you so much. I talk about putting on the pressure now. Let me see if, oops. I am sharing, am I? You get that?
Yes.
Okay.
I'm seeing
Yeah.
your speaker notes.
You can see?
Yes, I can see your speaker notes as well, though.
Oh, well, that's wrong. How's
Perfect.
that?
Perfect.
Okay. Okay. Yeah, this is a topic that is not one of my favorites, and I've been kind of dreading getting into this. But just some things over the past month or two have really kind of come to the forefront, and I said, let's go ahead and talk a little bit about lab regulations. And the reason is we are occasionally asked to do things. I think probably a lot of physicians and nurses kind of gripe about the laboratory being very set in their ways. This is the way we're going to do things, and we don't take to changes very well. And then there are some issues coming up. I'll talk about lab developed tests that really everybody in your laboratory is freaking out about lab developed tests right now. So who regulates the clinical laboratory? It really boils down to the regulatory aspect is going to be these two groups right here. The clinical laboratory improvement amendments regulated by CMS, and the Food and Drug Administration. So we obviously have to follow their rules. That's the law. This is written down in very legalese language in the Code of Federal Regulations, all the rules that laboratories have to follow. But we also get input from other groups as well. I'm putting these here, I'm writing these as the guidelines, the recommendations. You might look at that and say, well, what are you talking about? CDC is a federal agency. CDC really is regulatory in nature. But it's not really. CDC really going to be is providing a lot of good guidelines for us, recommendations on how we should do things. There's certainly a public health aspect to their regulatory authority, but when it comes to clinical laboratories, they really serve as a support system for the regulation of CLIA and FDA. CLSI, you hear us harp about CLSI all the time, looking at the rules and following their procedures and everything. But CLSI is strictly a guideline-based organization. They don't have any regulatory authority whatsoever. They can't tell us what to do. They tell us what we should do, but it's completely our prerogative of whether we want to follow their recommendations or not. And then we get to the accrediting body. So on the hospital-wide perspective, you got Joint Commission, you got DNV, and you got to follow their rules if you want to maintain your accreditation. They don't really get involved with laboratories too much because most of our hospital laboratories are going to be accredited by the College of American Pathologists. And they kind of deem that if you go through and you got your CAP accreditation, then the laboratory is functioning pretty well. So when it comes down to who really sets the rules that our laboratory has to follow, it really kind of boils down to CAP. In order for us to maintain that accreditation, CAP is going to make sure that if you meet all the requirements for accreditation, then you're going to be following all the CLIA laws and all the FDA laws. That's pretty much set in stone. So the chief body that gives us the rules that we have to live by is really going to be College of American Pathologists for most laboratories. I'm sorry about this picture and everything, but I just wanted to point out one of my frustrations when we start talking about laboratory regulations is every time I go to a presentation given by somebody who's talking about regulatory rules, standards, you always have a slide like this where they say, look, this is just what I'm thinking. These are my opinions and my suggestions. you're on your own as far as making decisions for your own laboratory. We lab people don't like that. We like things in black and white. I have been to several presentations given by CMS people, by the CLIA folks, by FDA and by CAP, and they all give a disclaimer like this. And I'm like, well, if you guys aren't willing to write it down on paper and say, this is the rule, this is what you have to do, then how are we supposed to know what's supposed to go on? So there's a little bit of confusion there. And part of it is because nobody wants to, jump in there and actually give you a definitive answer some of the times. Let's talk about FDA then and how they classify laboratory tests. First of all, any of the stuff that we're going to use in our laboratory, any laboratory tests are going to fall into the broad general category of IVD, in vitro diagnostics. These are reagents, instruments, systems that are intended for use in diagnosis of disease. That's what we use in the laboratory. FDA will classify those as class 1, 2, or 3, depending upon the amount of risk that's involved with that. And that applies across the board in things that are non-laboratory as well. Classified as 1, 2, and 3. But it's not just IVDs. There are some other things. I'll talk a little bit about some things that are called ASRs, analyte-specific reagents. This could be a company, for example, that's producing antibodies that react with salmonella. Okay, that is an analyte-specific reagent. It's not a test in itself. It's just a reagent that you could use for a test. If I produced a set of primers that could be used for a PCR assay to detect varicella zoster virus, that could be an analyte-specific reagent. I'm not making it for the specific purpose of doing an in vitro diagnostic test, but it is a reagent that could be used for that. And laboratories can take those and create their own laboratory developed test, LDTs. We'll talk a lot about LDTs coming up here in a bit here. This basically would be a test that a laboratory can get the reagents for, produce the test within their own laboratory, and run it within their own laboratory. We'll talk about that a bit more later. You'll also see some things that are labeled as research use only, or IUO is investigational use only. Those two terms seem like there's a lot of overlap, and there kind of is. FDA does make the distinction between those. A research use only product would be something where the test is in development. You're basically providing that to laboratories so that they can develop their own test. There's not a protocol, there's not a specific test that's done at this point. Investigational use only is going to be there's a test, you provide it to laboratories so that they can evaluate it. It's not on the market yet as an IVD. So there's a little bit of a distinction between those two things. But a laboratory can also take those and use them to make their own laboratory test if they so choose to do so. We got to throw EUA in here. EUA has always been just a little bitty corner down in the bottom, emergency use authorization. We never really thought of emergency use authorization much. We got a little bit of it when Zika came along. There were some EUA tests for Zika. And then, of course, when COVID came along, there was all kinds of emergency use tests that are out there. So these are tests that are being put on the market without going through the full FDA approval process, just because there's a need, there's a public health need for them. All those EUAs, eventually that emergency use authorization is going to expire, and they're going to have to eventually go through the full FDA process. Speaking of that FDA process, all these IVDs end up getting classified into a few different groups. They can be FDA approved, FDA cleared, or exempt. A lot of IVDs are exempt. That means FDA looks at them and says, the safety here is not a concern. There's no real reason to question the effectiveness of the test or whatever it happens to be. So we're not going to require a full documentation. So most of the things really that we use in microbiology are FDA exempt. You look at things like our media tests that we use to identify bacteria, a lot of those are going to be FDA exempt. FDA cleared versus FDA approved will depend upon the risk that's involved and how much documentation is required to be submitted before the FDA is going to make a decision. The other thing we need to add in here is all the non-commercial methods. FDA regulates commercial methods. They do not regulate non-commercial methods. So, for example, nobody has FDA cleared or FDA approved a urine culture. Now, some of the things that we use in the process of that urine culture will be FDA cleared or FDA exempt, but a urine culture itself is a non-commercial method, so it's not going on the market. It's in this black box here. So let's kind of make a comparison here, and this is going to be one of my gripes here. So you'll just have to put up with me griping a little bit about this. FDA not just approves lab tests, of course, they also approve drugs as well. And part of the process is not just approving the drug or the lab test, but actually approving the documentation as well. So with drugs, you're going to have this document of prescribing information. Excuse me. See, my gripe about it, so I end up choking myself. This prescribing information in this document actually goes through FDA approval as well. Just like with a lab test, we're going to have the instructions for use that will also have gone through FDA approval. Sorry. Okay. So it's important to look at the indications. So if you look at the drugs, for example, this is part of the prescribing information for ceftaroline. And it says, here are the indications. These are the FDA-approved indications: acute bacterial skin and skin structure infections, community-acquired bacterial pneumonia. This is the test that we use for HIV viral load. And just like a drug, there's going to be intended use. There's going to be certain indications that it's allowed for. It's got to be used on that instrument, intended for clinical management of HIV-infected individuals. You look down here, not intended for screening blood products, tissue, or organ donors, and not intended as an aid in diagnosis. In other words, the intent of this assay is to monitor people that you know have HIV. It is not to diagnose HIV in the first place. And this is where I run into a little bit of a gripe here with me. There's a bit of a disconnect between how doctors are viewed and how laboratories are viewed. So here's a quote from the FDA's website. From the FDA perspective, once the FDA approves a drug, healthcare providers generally may prescribe the drug for an unapproved use when they judge that it's medically appropriate for their patient. So doctors are allowed to use judgment in deciding whether a drug is appropriate or not. Here's a quote from CAP. From a regulatory standpoint, any susceptibility testing that is not FDA cleared or approved must be validated prior to releasing patient results. There are no exceptions. There are no exceptions. Laboratories are not allowed to go off-label unless things have been validated. So if we go back to this right here, let's look at ceftaroline. If a doctor wanted to prescribe ceftaroline for hospital-acquired pneumonia or pyelonephritis or sepsis, that's fine. They're okay to do that. But from a lab perspective, excuse me, I've been talking too much here. From a lab perspective, there's no FDA indication for those conditions. Therefore, there are no FDA breakpoints, and therefore there's no FDA-approved susceptibility test. Excuse me. So labs are not allowed to actually do susceptibility testing for these indications unless they have validated it in-house. Look at the HIV test. Sorry, I'm gonna have to give myself something mint here to clear my throat out. If a doctor wanted to order an HIV viral load assay to actually diagnose HIV, to show that the patient has HIV, they're welcome to do that. But the lab is not allowed to do the test until they have validated it for that purpose. So there's a little bit of a disconnect here between what physicians are expected to do and what laboratories are officially allowed to do. This is an extremely simplified version here of what a company would do if they wanted to develop a lab test. So let's take a look at this. Sorry. Medical world says we need a PCR assay for pneumocystis. So what this company does is they do some research, they figure out primers and probes, they design the assay, and then they will do a lot of in-house work. Sorry. To optimize that assay, to get it working just right. Then the best thing to do would be to take that assay and distribute it out to laboratories that you contract around the country. And have them help to develop data, to test it, to spike specimens, real patient specimens, to evaluate that assay. And then collect all that data, write all that data up, including all the specifics about how well the test works, and all the specifics about what it can be used for, the indications, what specimens, etc., and submit all of that to the FDA. Okay, very simple, believe me, very simple. But before a lab can actually do the test or the clinical laboratory, you have to do either verification or validation. And I wanted to talk about those two terms because there's a lot of confusion over exactly what they mean. And laboratory people have kind of done that to themselves. And that different organizations have used, excuse me, have used different definitions for those two terms. No, I'm sorry. I'm getting over a cold here. So before a lab, sorry, before a lab can offer a test, we need to do either a verification or a validation. So a verification means I'm going to take a test that has been FDA approved or FDA cleared, and I'm going to do it the way the manufacturer says to do it. I'm going to use their protocol. I'm going to use their specimen sources for the indications that they describe. And so what I need to be able to do before I can start doing that test is demonstrate accuracy, demonstrate that I can get the appropriate sensitivity and specificity as is defined in the instructions for use. Precision means reproducibility. I can do it without coffee. Precision means I can take the same specimen. Sorry, I'm gonna just drink some more here. I'm gonna take the same specimen, run it multiple times over a course of a few days, and get the same results. Reportable range is usually pretty simple for micro. Sorry. The reportable range is typically going to be positive or negative. It's a little more complicated when you get to chemistry tests. And the reference value is usually pretty simple too. For microbiology, it's usually negative. There's nothing there. Now, a validation is different. It's going to be more involved. A validation means I'm doing something off-label. Sorry, I would mute myself, but I can't figure out how to do that. Um, a validation means I'm doing something off-label here. I'm doing a modification of the assay. I'm doing something here that is not FDA cleared or FDA approved. I still have to show accuracy. I still have to show reproducibility, but I'll also need to show the limit of detection, how little can be there, and I'll still stand a good chance of detecting it, and a little specificity, which means does it cross-react? For example, if I'm going to do, excuse me, a PCR for Neisseria gonorrhea, I need to make sure that it doesn't react with a whole bunch of other species of normal flora Neisseria. I need to be able to test a whole bunch of other bugs that may be in those specimens, and a whole bunch of other things that may be necessary depending on the specific test. Now, I want to kind of point out that as far as I look at it, validation really comes in two different flavors. There's a technical validation and a clinical validation. Technical validation, it means I've changed some aspect. Excuse me. I'm using a new transport medium, for example. All I need to do is show what effect that has on sensitivity, specificity, limit of detection. This is something that can be done completely in the lab. Can I do it? Yeah, I can do it. It might cost me a lot of money. It might cost a lot of time, but it's generally possible to do. Sometimes I need to do a clinical validation. For example, I've added a new indication or a new specimen type. Does a positive or negative result indicate what we think it does? For example, if I add a different specimen type, is the bug that I'm looking for actually going to be in that specimen? Does a negative result really mean that the patient is negative? That's a very difficult validation to do. And most of the time, a laboratory is really not going to be able to do that. So it kind of depends upon what you want to see. For example, if I got a PCR for adenovirus, I apologize for all the caffeine. I thought I was going to be able to get through this. PCR for adenovirus is FDA cleared in viral transport medium. But if I want to use e-swabs, that would be a technical validation. It would be pretty simple, pretty straightforward for us to do that. What about this one down here? PCR for flu, RSV, and COVID. FDA cleared for nasal and nasopharyngeal swabs, but we want to test saliva. That's going to be a clinical validation initially, because I would need to be able to demonstrate that if you are infected with one of those viruses, then they can be found in the saliva. Then once I can see that they've been found in the saliva, then we need to be able to show that we can actually detect it in the saliva. What would our limit of detection be? How do we collect that saliva? How do we process saliva, et cetera? So that's a much bigger deal. Let me skip through here because I'm getting, okay. Let me go back to this here. So this happened to me just a couple months ago. A physician requested that we run a respiratory pathogen panel on a spinal fluid specimen. Obviously off-label, not allowed to do that. So you would look at that and say, well, no, you can't do that because that would require a rather extensive clinical validation and technical validation. But actually, CAP lets us have a pass here on something like this. Sure, On a one-time basis, I can do it. I can put a disclaimer in saying it's an unvalidated specimen type, but I can go ahead and run it. Obviously, I would have to have a discussion with that physician to make sure we're looking for something that we'd expect to be seen in a spinal fluid. So, yes, I can do it on a one-time basis, on a two-time basis, maybe. But I'm not allowed to make a habit of it. If we get to the point where people are requesting it, then I'm going to have to say, no, we can't do that without a validation. So this doctor called me, asked for it. We discussed it. I said, okay, let's do it. We ran it. And a week later, a different physician called and requested that we do a spinal fluid on a respiratory pathogen panel. And I said, no, sorry, but we can't get in the habit of doing that unless we validated it. Let me talk briefly about laboratory developed tests, because this is as soon as I think I'm good. Laboratory developed tests. These are tests that are intended for clinical use, but are designed, manufactured, and used within a single laboratory. So here's the plan of how this is supposed to work. In my lab, for example, we decided that we need to have a PCR that will detect anaplasma and Ehrlichia. We can go about designing this. I can either design the probes from scratch, or we can purchase analyte-specific reagents. So we get those. We put it together in a procedure. We evaluate the assay. We do whatever validation we feel is necessary in-house. And then I can offer that assay in our laboratory. The evaluation is done completely in our own laboratory. We do not send it out to other laboratories to assist us with that. And I don't submit it to FDA for approval. Okay, once I've got my validation and I signed off by my medical director, we can run it. All right. That's the way a laboratory developed test typically works. Now, there's lots of different degrees here. We just talked about this one right here, a brand new novel test. But any of this is also considered a laboratory developed test, including just modified an FDA cleared IVD would be considered a laboratory developed test. Now, this is what FDA kind of their stance on it. When they first talked about LDTs back in 1976, these were typically going to be a small number of laboratories that really did this. Excuse me, assays that I really am sorry about this. I should have had a backup here. Most LDTs were low risk, small volumes. And so the FDA said, we're not going to enforce these. We're not going to enforce what we call the applicable requirements. Last month, the FDA came out with a new rule. And this is what they're saying now. LDTs have changed. Used more widely, used for a larger, more diverse population. There are large reference laboratories that will create an LDT and not just run it, they'll run it in their own lab, but they'll accept specimens from all over the country. A lot of the assays that we send to our reference laboratories are LDTs that were made at that laboratory. And so they have decided that they are going to phase out enforcement discretion and start treating laboratories as test manufacturers. So the way this flow chart changes is we still come down to here. Excuse me, we are still expected to evaluate in our own laboratory. We don't send it out to other labs. But But now we're going to have to submit to the FDA for approval in between here. That's what's coming up. Where does everybody stand on this? This is what the FDA says. This is needed to ensure safety and efficacy of complicated lab tests, many of which are marketed to labs nationwide. So they're not really these small little tests that are offered in low volumes within a particular laboratory anymore. If I look at that, I'd say, you know what? They have a good point. Okay. But if you look at pretty much all the laboratory groups that are out there, they're all freaking out. They'll say, we already have CLIA rules and we have CAP standards that ensure the safety. FDA is not going to be able to assure the safety any more than what we've already got. FDA's plan is going to be a huge burden on laboratories. How is a laboratory going to collect all that data within their own laboratory and then submit it in a reasonable amount of time and a reasonable amount of cost in order to develop tests that are needed now? The reason that you need an LDT is because it's not commercially practical for a company to put that kind of money into it. All right, but now laboratories will be required to do that. They get right down to it. FDA doesn't have the people to evaluate all that anyway. So you look at the laboratory stuff and you say, okay, they have a good point. Everybody's got a good point here. But we're going to have to wait and see how it plays out. I'm going to skip this since I'm already over time and since I'm choking to death here. When is it going to go into effect? Officially, it went into effect in May. It will be fully phased in over a 4-year period. This is what's probably going to happen, though. The lawsuits have already been filed, and it will probably be decided in the court at some point. My point in saying all of this is that you're going to see your laboratory kind of freaking out a little bit over the next several years, trying to figure out, are we capable now of making our own laboratory developed tests in rapid response to something like COVID, for example? You look at all the early COVID tests that were done, those were all laboratory developed tests. Are we going to be able to do that? Anyway, clinical laboratories have got to verify or validate tests before you offer them for clinical use. Generally speaking, we're not allowed to deviate from any of this without doing a validation first. So laboratories have to be a little intransigent when they get requests from physicians and nurses. It's all really very gray. I'd like to be able to say that there's definite black and white rules here, but there's not really. And like I said, the new LDT rule is going to be a hot topic for several years. And I'm going to quit there and try to not choke to death. I do apologize. I was getting over this cold last week and I thought I was good.
Well, thanks so much, Dr. Junkins. That was very informative. I typically send something to the lab and the magic happens and I get a result. And I wasn't even aware of all of these guidelines and accreditation and all of these things. Thanks so much. Does anyone have any questions for Dr. Junkins? I'm not seeing anything, Dr. Junkins. All right. We'll end here. Thank you all so much for joining us. Thank you, Dr. Junkins.
Thank you. Apologize for the coughing.